Have you ever wondered how proteins are made inside your body? The answer lies in a tiny yet mighty nanomachine called the ribosome. Proteins are central to life. They take part in almost every biological process. Without them, life as we know it would not exist. Their building blocks are amino acids, 21 different kinds in humans. These link together to form a linear chain called a polypeptide, which folds into a functional protein. There are almost endless ways to assemble these amino acids into different chains, and each unique sequence creates a protein with a different role. Our body produces hundreds of thousands of different proteins. But how do cells know the correct order in which to assemble amino acids? The instructions are stored in our DNA, inside our genes. These genes are first copied into molecules called messenger RNAs, or mRNAs for short. mRNAs carry the code that specifies the order of amino acids in the future protein. The actual synthesis happens in protein factories called ribosomes. These are highly specialized molecular machines that read the genetic code in mRNA and translate it into proteins. Every cell in our body contains millions of ribosomes, essential for all life. But what do these fascinating machines look like, and how do they work? Thanks to the pioneering work of Venkatraman Ramakrishnan, Ada Yonath, and Thomas Steitz, we now have a detailed picture of the ribosome. Their heroic efforts earned them the Nobel Prize in Chemistry in 2009 for studies of the structure and function of the ribosome. The scaffold of the ribosome is made of ribosomal RNA, or rRNA. These rRNAs fold into complex three-dimensional shapes and together with many proteins form the large and small subunits of the ribosome. The ribosome assembles on the mRNA and reads its sequence. Guiding transfer RNAs, tRNAs shown here in red, which bring the correct amino acids. Step by step, amino acids are joined together to build the polypeptide chain, like an assembly line. Remarkably, the catalytic function of linking amino acids is carried out by the ribosomal RNA itself, making the ribosome a so-called ribozyme. This and other ribozymes support the idea that life on Earth began with RNA. By unlocking the ribosome's structure, scientists revealed the molecular details of how genetic information becomes protein. This discovery didn't just solve a scientific puzzle, it opened the door to new medicines, from antibiotics to cancer therapies. From decoding the secrets of life to revolutionizing medicine, the ribosome's story is far from over. Here's to the tiny machines that make life possible. Fundamental discoveries about RNA continue today. Learn more on the Molecule Cosmos RNA website and watch the other videos in this series.